about some of the soil microbial work we've been doing in nurseries, and this is really focused on ectomycorrhizal associations. So first I'll just start off with a bit of a background around why we were looking at nursery management as a potential factor. Well, if you can imagine your tree out in your forest, it has any number of factors that influence its productivity and its health. The genetics of the tree, chemical use such as fertilizers or maybe copper as a fungicide, the way in which the site is prepared prior to planting, how you disrupt potential nutrient supplies from harvest residues, whether or not what sort of work you do on the site. Also bacterial and fungal interactions at the forest site itself. Obviously climate has a big influence on tree health and productivity. And again, silvicultural decisions around stocking rates, thinning rates, whether or not you prune. But sitting in amongst this is also the impact of nursery management in handling practices. And these are important because they influence the state of the tree stock as it leaves the nursery gate. And we know that this has important implications for the growth in the plantation because of its impact on initial survival rates, initial growth rates, and also the potential to cause something called new ground syndrome. And I'll define that last one a little bit later on in the talk. Now, nursery research has already accomplished quite a lot. Studies exploring how to improve the survival rates of Pinus radiata planting back in the sort of the 50s and the 60s have done a lot of work. Initially, we had survival rates of perhaps 50%. So if you wanted 1,000 stems per hectare, you'd plant 2,000 stems and you'd wait for half of them to die. But a lot of physical practices such as undercutting and wrenching and topping have improved seedling condition. And we've also developed targets for seedling diameter, which basically say, you know, if you're over a certain, if you're like over 5 millimeters, your chances of survival are that much higher. And a lot of this, these gains have been also underpinned through the better use of fertilizers and fungicides, fertilizers to increase growth rates to get to those seedling diameter targets, but also fungicides have become increasingly important because monoculture tree nurseries have a lot of problems with disease. And again, advances in just physical handling practices, the logistics side of it, systems involving plastic bags to retain moisture, the use of white planting boxes and storage pods that reflect heat, and again, a system of planter box holders and bags, which basically mean you're able to keep that seedling in a reasonably protected environment up until basically right up until the moment where it's actually planted back in the ground again. So these have all made a really big difference. But one of the areas that Scion has done a lot of work with in the past has been biological interactions. And indeed, some of the first nursery work, and indeed the first work at all that Scion did, was with mycorrhiza. Now, I talked about this a little bit last week, but I'll just cover it again. So ectomycorrhizal fungi are just fundamental to the ability for conifers to capture resources from soil. If you can imagine that the soil volume that this seedling can explore and can exploit, just by looking at the roots alone, it's quite small compared to the soil volume that the ectomycorrhizal network allows that tree to access. So, you know, a huge shift in the ability of that seedling to acquire resources if it has an active ectomycorrhizal fungal network supporting it. Now, we had an early success story in New Zealand with ectomycorrhizal research that actually predates the formation of Scion in its current form. Back in the 1930s, when Douglas fir was first introduced to plantations in the Nelson region, there were significant problems. The seedlings would be bought in and planted, and then they'd either not do much and turn yellow for a couple of years or perhaps struggle along. And this was basically chlorosis. They just weren't getting nitrogen and other nutrients out of the soil. So the forest ranger, a chap called Ari Lawrence, believed that this was occurring due to a lack of mycorrhizal fungi on the seedlings. And this is something called new ground syndrome, where you introduce a tree or a plant to an area that doesn't have the symbiotes it needs, and the production facility that's producing those seedlings doesn't equip it with those symbionts when it's leaving the nursery gate. So in the high nutrient environment of the nursery, the Douglas fir was doing okay, but when it was transplanted to a harsher site without the symbionts it needed, those mycorrhizal fungi, it just wasn't able to deal with those conditions. And because there had never been Douglas fir at this site before, the pool of those mycorrhiza had never developed. So to solve the problem, he hopped in his truck and he drove from Nelson to Hamner Springs to collect litter from a successful Douglas fir plantation that had developed in that area. Now, currently the drive from Hamner Springs to Nelson is about 4.5 hours going through multiple mountain ranges. And I'm terrified to think what this would have actually looked like in the 1930s, but he was committed to his idea, so he hopped in his truck and off he went, collected the litter, and then drove back to Nelson. And in the end, this was ultimately successful. The leaf litter that he spread around the existing plantations removed the chlorosis, the trees recovered and started to grow well, plus the seedlings that he introduced into the area were also able to access and form associations with those mycorrhiza. However, the only problem is that this work was never really recorded, and we've only basically stumbled across the records of this work something like 10 to 5 years ago. However, we did have another early success with, well, as a consequence of that story not being particularly well known, we had the exact same problem again with Douglas fir, again, this time in the 1950s in the Otago region. But only by this point, Scion had been formed and an extensive study was able to be undertaken. But it ended up being exactly the same story, essentially, that the problems with chlorosis and the lack of growth were tracked down to a lack of mycorrhiza in one of the nurseries that was supplying the stock to the plantings. So on that little panel over there, you can see seedling A, it's just got its bare roots, whereas seedling B, you can see those clusters coming through on the roots. Those are the mycorrhizal fungi that once that tree is planted will form that dense network of hyphae that allows the tree to capture many more nutrients and resources from the soil. And again, they were able to solve the problem through the exact same method. They took forest litter, which contained those ectomycorrhizal fungi from a successful Douglas fir plantation, spread it around the nursery, spread it into the plantations that they'd already established, and the trees recovered, and the new seedlings coming from this nursery had the right mycorrhiza. And this time, everything was fully documented. And just to give you an idea of the extent of the problem, this new ground syndrome with Douglas fir still occurs, well, was still an issue perhaps even 20 years ago. So here I have my colleague, Murray Davis. He is holding 2 Douglas fir seedlings. They had been planted 2 years previously at a relatively harsh site in a part of New Zealand called Tekapo in the Mackenzie country. And the one that you can't see didn't have ectomycorrhiza on it when it was planted. So the big one, it had the right ectomycorrhiza. It was able to adapt to the site and thrive and establish itself and develop, whereas the other one basically spent 2 years not dying. It's probably has a lifespan in months. Well, admittedly, it was pulled out of the ground for this comparison, but the similar trees like that, they all perished quite rapidly after this photo was taken. So again, that's just the importance of getting the right mycorrhiza in place for species that are sensitive to mycorrhiza. Now, moving on to far more recent research. Well, all of the work that was done in New Zealand up to the early 80s around improving nursery practice positioned New Zealand as a real world leader in applied nursery research to make differences that affected not just practice in the nursery, but played out in the forest as well. So naturally, of course, the government looked at this and decided to stop all funding because we'd reached the perfect point. And as a consequence, there's been no significant research for a good 30 years after that point in the early 80s. There is actually another reason for that I'll come to later. But as a consequence of the lack of systematic nursery research, but the increasing pressures being placed on nursery managers to deliver crop, the result was an intensification in chemical use. The main source of advice for nursery managers during this time period was chemical reps basically, and their solution for most things was, well, just use more chemical. So as a consequence, the rates of fertilizer and fungicide use within tree seedling nurseries increased dramatically during this period. Fortunately, in 2009, a combination of some new funding, but also a big uptake in industry interest in actually exploring what was going on, allowed research in this area to be revitalized. And we decided to sort of look at three key areas. The first was, what was the impact of all this increased chemical use in the nursery? We knew what it was doing to the seedlings within the nursery, but what were the legacy effects in the forest? How was the change in nutrient and chemical application in the nursery affecting the performance of those seedlings in the forest? The second area that we looked at was, what are the implications for the increased chemical use on the ectomycorrhiza that we know were very important, and how is that influencing the formation of those associations within the nursery? And the third part was then understanding, well, if the ectomycorrhiza in the nursery have been changed or are affected by chemical use, what are the flow-on implications for the performance of those seedlings in the forest as well? So to explore this, we set up our first mycorrhizal study for about a good 30 years at Tenai Nursery. It was a fairly simple design. We basically what we did was we compared standard rates of fertilizer and fungicide with 25% reductions and 25% increases. So we basically had 3 treatments for fertilizer, 3 treatments for fungicide, and then we looked at all possible combinations. So we had 9 treatment levels plus some control treatments as well. Now, after 9 months of treatments, which included multiple applications of fungicide, multiple applications of fertilizer, we assessed seedling growth, seedling nutrition, and we also looked at the ectomycorrhizal associations that had formed on those seedlings during their time in the nursery. And to do this, we conducted what was, and I think still is, the largest ever study of mycorrhizal root tips. We identified the taxa of 150,000 individual root tips. So there was myself and a team working on this. I firmly recommend no one ever tries this because it was incredibly painstaking and took forever, but it did produce just an enormous quantity of data, which has become incredibly valuable. So one of the first things we found that really surprised us, that the chemical use or variation in chemical use had no impact whatsoever on total mycorrhization. That is the number of mycorrhizal fungi that were present on any given seedling. But what we did find was that the composition of the community of ectomycorrhiza that on the seedlings was incredibly sensitive to the different chemicals. So we weren't affecting the total amount, but we were affecting the individual species that made up that population. And one of the things we found in particular was that the species Rhizopogon rubescens was exceedingly sensitive. So basically, the more chemicals you added, the less you had of Rhizopogon, and it was instead replaced by Wilcoxinia and Hebeloma. And it was sensitive to both fungicide treatments, which were used to protect the crop from disease, and fertilizer addition, which was used to enhance their growth. But interestingly, we also found that reducing the fungicide had no impact on the health of the crop whatsoever. So the next step was to look at how this relates to the metrics, the productivity metrics of the crop. So one of the first things that was actually physically observable as the trial went on, was that the trees, or the seedlings that received less fungicide, were actually growing bigger. And when we did our end of rotation assessment, we ended up with a fairly obvious result that you can see here in this figure, that the less fungicide the trees were exposed to, the bigger they were when they left the nursery gate. But we also picked up other impacts. When we looked at the nutrition, we found that the more Rhizopogon rubescens a given seedling had, the more nitrogen, magnesium, sulfur, and manganese it was also likely to contain as well. And by comparison, all other mycorrhiza that we were looking at were essentially unrelated to nutrition, or indeed had negative correlations with nutrition. Now, this isn't to say that these other mycorrhiza were bad for the tree or they were pulling nutrients out. It's just that any root space that was occupied by a mycorrhiza other than rhizopogon, it just wasn't doing as good a job. So therefore, it was it was just an effect that if you had another mycorrhiza, you didn't have rhizopogon, so therefore your nutrition wasn't as good. Anyway, the full details of the study are published. We did this a few years back, so the detail on that is accessible through this publication. Now, the 10i trial, that first one that we did provide useful data, but it's limited simply because it was one cohort of seedlings in one nursery in 1 year. So to basically see if we could replicate our results, we established another trial at the Scion nursery in 2014. And in this case, we extended the boundaries. We pushed our changes in chemical use to 50% reductions, or in the case of fertilizer, a 50% increase. We didn't do a 50% increase in fungicide because we could just see no particular point in that. Another reason why we chose the Scion nursery was that this nursery is quite old. It's been producing Pinus radiata for at least 75 years. And as a consequence, it's built up quite a pathogen load. We were expecting that here we might see reductions in fungicide would have a negative implication in terms of greater disease rates. However, we basically saw the exact same results that we achieved with the 10i trial. The more chemical you used, the less rhizopogon that you saw. We didn't see any reductions in productivity through reducing our chemical use. And again, we had no impact on disease rates through reducing fungicide use. And this panel here just gives you an example of the reduction in rhizopogon that we saw with increasing fertilizer use. So again, all nurseries are different. There was a slightly different mix of ectomycorrhiza present at this site, but still dominated by rhizopogon, with Wilcoxinia as the second most common species. So that trend was still the same. But again, you can see the relative abundance of rhizopogon decreasing with increasing fertilizer addition. By this stage, our results were starting to attract a little bit of attention, and through discussions with Arbogen, which is a sort of an entity that produces across multiple nurseries in New Zealand around about 40% of the Pinus radiata stock, we were able to move to operational scales. So basically I was provided access to about 2.5 kilometres of seedbed in 2015, which allowed me to treat around about 250,000 seedlings. And the key thing we decided to undertake in this trial was to see if we could replicate what we had achieved at research sort of scale plots. Could we do that at operational scales? So the two questions that we posed ourselves were could we replicate the maintenance of seedling growth by using fewer nutrients, which is what we'd observed through our fertilizer reductions, and could we replicate the maintenance of seedling health while at the same time reducing fungicide use by 50%. So integrating the fertilizer practices was quite simple. That was just a case of applying less or applying different products. Whereas for the fungicide, reducing fungicide use by 50%, again, we took a pretty simple pathway. If you can imagine that the nurseries are established in long beds, what we did instead of applying fungicide to every single bed, we applied it to every second bed as indicated by the orange highlights in that panel on the bottom right. So basically the tractor that was applying the fungicide, instead of going up and down every single bed within a given bay, it would only apply fungicide to every second bed within the bay. And again, in all honesty, the results that we got out of our operational scale trials were even stronger than what we saw in the research trials. So here is an example of the fungicide reduction trial. So the left-hand side of the image shows one of the beds that received fungicide, and the right-hand side shows a bed that received no fungicide. And again, this is after about 9 months of these differences in treatments. As you continue over on the right, the strips continue. So it was just strips of fungicide, no fungicide, fungicide, no fungicide. And again, this covered kilometers of seed bed by the time we were finished. And so the net outcome was absolutely no reduction, no change in disease incidence throughout the entire nursery. And again, we tried to stack the deck because this trial was located in the single most disease-prone part of this relatively old and extensive nursery. But nothing came through again. So in terms of the growth, however, we saw increased gains in seedling diameters with less fungicide, as we'd observed before. But in this time, because of how we described how we set up the experiment, it was a little different. So I'll define the standard treatment as just the normal run-of-the-mill treatment. So basically, the bed that the seedling was sampled from received fungicide, and the adjacent beds both received fungicide. The received treatment, those seedlings were in beds that did receive fungicide, but neither of the neighboring beds received fungicide. And the adjacent treatment, wherein these seedlings received no fungicide for the 9-month period, but the beds on either side of them did receive fungicide. But as you can see, with diminishing exposure, you're having an increasing gain in root collar diameter leaving the nursery gate. And again, that's quite an important finding. Going beyond that, we also explored the impact of the fungicide treatments on seedling nutrition at the point of harvesting when the seedlings were leaving the nursery gate. So we found a significant gain in boron with reduced fungicide use, no impact on nitrogen nutrition. We did see a reduction in phosphorus, and this is simply because the fungicide treatments tend to contain a lot of phosphorus. But anyway, regardless of this, both the phosphorus content in the received and adjacent treatments was still considered well above the sort of minimum criteria for a healthy seedling. However, considering other cations, we did see significant gains, zinc as an example, in the uptake of these nutrients with no fungicide, despite the fact that we were adding no additional fungicide. All we were doing was we were adding no additional zinc or boron. We were just restricting the amount of fungicide. So again, this was pointing to the fact that we had a different mycorrhizal community that was more active, and it was delivering more to the seedlings. And again, the work we did then to characterize these the ectomycorrhizal communities leaving the nursery gate showed us that we had created different ectomycorrhizal communities, and that was paying off in terms of differences in seedling qualities. Well, that's all well and good in terms of activity within the nursery, but the real impact comes from how this plays out in the forest. Do any of these differences that we have leaving the nursery gate actually matter to forest productivity when you take that stock out and put it in the forest for several years? So to explore this, from each one of these trials I've described, we did outplanting work. So from the very first trial conducted at the Tenai nursery site, and that started in 2009, we took around 2,500 seedlings from within that trial, and we planted them out at what is a relatively typical forest site. During the life of the trial, we have routinely assessed mortality and growth, and also during the initial stage of the trial, we collected ectomycorrhizal samples to see how the ectomycorrhiza that were present on the seedlings when planted, has that changed, and how quickly has that changed. Now, we found that within 2 years, new mycorrhiza that were present at the site, but not in the nursery environment, did start to appear and capture more root area. But we also saw significant legacy effects from the nursery treatment starting to become apparent. And this manifests as increased survival rates and increased growth rates. Now, when we did an assessment in 2006, we started to see some quite significant results. And these included a legacy effect of reduced fungicide manifest as increased DBH and significant gains in the basal area of the plots of the trees that had received less fungicide in the nursery. Now, I'd stress there have been no other treatments once these trees were planted. They were simply taken away from the nursery, planted out at the site, and everything was just left to see what happened next. So what we've basically seen is that we've done our best to try and isolate other effects across the site that could have been causing these, but they really have come to nothing. What we think the key driver of this legacy effect is that the reduced fungicide treatment created a better ectomycorrhizal community on the stock when it left the nursery gate, basically more rhizopogon, and that provided a head start for these trees that has continued to persist now 6 years later, and we've now got measurements that go up to 10 years later now that again show that there is an ongoing legacy effect. But again, the issue with the Tenai trial and the outplanting site is that it was still only one crop of seedlings going to one location. So maybe we were just very lucky. So to assess that, we took about 10,000 seedlings from the Scion trial that was established in 2014, and we established those across 5 other locations around New Zealand. Oh, sorry. And to date, we have seen some variation in responses. I'm quite happy to say that we have got our strongest response at that original site in Kaingaroa. But across the board, we have seen positive legacy effects from reduced fungicide use at a number of these sites, which is manifest as either better survival rates or faster growth. And probably more importantly, at none of these sites have we seen decreases in fungicide produce a worse quality crop. It's either been the same or improved. And actually that variation across sites gave us another idea to do another set of field trials as well. So this time we set up another 46 sites using the 2.5 million trees that we had available to us from the first operational trial at the Aberdeen nursery. So in this study, we took a bit of a different approach. We took basically 4 different recipes for producing stock that we'd created or deployed at the Aberdeen site. We took a standard seedling, let's call that treatment A or recipe A. So that was produced just through the normal way that the seedlings would have been raised in that year. And we compared that to 3 other recipes that we had tested, which involved reductions in fungicide or alterations generally, reductions in fertilizer use. And all of those 3 other recipes were better than the standard one. But we had achieved a better quality seedling through different pathways. And as a consequence, the mycorrhizal community on those 3 improved types of seedlings were also different as well. So this gave us basically 3 seedlings that were better, all with different ectomycorrhiza on them to some degree. And we deployed these across 46 different sites to provide an opportunity to figure out, was it actually possible to manipulate the mycorrhizal community to make a seedling more suited to a particular site? So across these 46 sites that we established in 2016, we have a wealth of data on the sites, chemistry, climatic data, past history. And what we're trying to do with this is figure out, are there any key parameters of a set of sites which say recipe B is the best for this type of site? Recipe C is the best for these types of site, maybe based on pH, maybe based on nitrogen phosphorus ratios. So that's what we're exploring. Now these trials are very much in their early phases, but we are actually getting indications that there are opportunities to do this, that you can manipulate a seedling and manipulate its mycorrhiza to make it more or less suited to a particular area or a particular type of site properties. Now, however, we do need to step back from that sort of goal of producing site-specific seedlings, because we've got to be realistic to the scale of sort of, that management ectomycorrhiza matching to a site. It's never going to be feasible to try and target nursery management to suit a small area. And I mean, 20, 200, or even 2,000 hectares is probably unrealistic. But what we do think is that there is opportunity to do better with our nursery management and our targeting of nursery management at larger scales. And a good example is Kaingaroa Forest. So as I talked about last week, Kaingaroa Forest, it's 186,000 hectares of contiguous forest. It's populated by seedlings that come from Tenai Nursery. Now, if you consider the properties at North Kaingaroa, Central and South Kaingaroa, they're all very different. The soil types are quite different. The climate is exceedingly different. Altitude and temperature regimes are quite distinct across these different areas. However, all of the stock that comes out from Tenai Nursery is treated the same. So what we're thinking, and what we'll hopefully be doing at some point in the future, is seeing, can we raise 10,000 seedlings to be better suited, can we raise seedlings to cover 10,000 hectares in Kaingaroa? In Northern Kaingaroa, here is your recipe for Northern Kaingaroa. Here is your recipe for perhaps providing seedlings for 20,000 hectares of Southern Kaingaroa. And here's a different recipe again for producing seedlings that's best suited to Central Kaingaroa. That's the sort of scale of operations that we think we might be able to work in. And but again, we are in the infancy of this particular work, but it's looking hopeful so far. So where are we at in terms of the risks and barriers to uptake of this work? Well, firstly, nursery managers are very conservative for a very good reason. Fungicide and fertilizer has underpinned reliable stock production. It reduces the risk of disease, and it allows seedlings to get to target specifications. So reducing the use of these products does create significant risk. And although we have had multiple successful demonstrations from reducing chemical use at operational scales, we've yet to see a significant change to fungicide use across the industry, even though we've just told nursery managers, go off, try this, and they know that they're getting better quality stock, but they don't want to deploy it across their whole nursery. And I fully understand that, and I'm never going to advocate using no fungicide. You need it in your back pocket just in case. But what we're trying to advocate is for better use of fungicide. Now, we have our results with altering fertilizer products, however, has seen some interest in changing practice at a large scale, but this is still very much, again, in its early days, but we are having an impact on nursery practice that's measurable right now in terms of nutrient management. So as it stands currently, I don't really have particular plans to launch new ectomycorrhizal trials. Right now, our effort is focused on tracking the existing field trials that we have so that we can quantify the full benefits for forest productivity. And then we can take that story to forest and nursery managers, because if forest managers want to see the benefits that we're able to create, they need to work with their nursery managers to take on some of that risk and to share some of that risk right at the start. Because that risk is very real, that if you reduce fungicide, you might not be delivering the number of seedlings that you're contracted to do. But if the forest manager wants that later improvement in performance, then they need to be adaptable to that risk as well. And now, just to sort of conclude, I'll tell another reason why we're not looking to push this into operational practice that quickly. We're taking a very softly approach. Back in the 1970s in New Zealand, a number of trials were done where mycorrhiza were introduced into sterilized hill country soil. And it produced some amazing results in terms of clover growth. We saw increases of up to 3,500% in dry matter growth after about 10 weeks. Now, this was massively promoted in New Zealand, widely reported, and it was promised as a new farming revolution that was going to be created by these mycorrhizal species. So a lot of investment went into the development of field trials to test this out in operational circumstances. And unfortunately, it produced absolutely nothing. When those mycorrhiza were introduced out into the wild, basically, they couldn't compete with the mycorrhiza that were already present on the sites at those soils. So after a lot of money and resources and a lot of reputations had gone into the importance and the impact that this was going to produce, there was a bit of grumpiness. And the consequence of that was that mycorrhizal research was basically considered to be ineffective by the government. So all mycorrhizal research across all sectors suffered deep funding cuts, and there was just no one was interested in it anymore. And this had a direct impact on the tree nursery research in New Zealand, because that was the next thing that was going to be studied. Mycorrhizal research was at the forefront of the next nursery research program. And even though the tree nursery mycorrhizal research had nothing to do with this agricultural research, it was cut and funding was taken away just because of the reputational damage done to mycorrhizal research. So as a consequence, I'm quite happy to be conservative about what we've done, and I'm quite happy with an 8% gain in basal area, and I'd be very suspicious of a 3,500% gain in basal area. So anyway, where do we come back to? Well, nursery research and the work that we've done with ectomycorrhiza, it isn't the end-all be-all. It's just another tool, just like silviculture, just like the manipulating the genetics of the tree, just like understanding climate, just like understanding silvicultural practices. It's another set of tools to get more from the forest. I think that in the last 10 years, we've caught up a lot of ground. We've made up a lot of ground on that 30-year research gap, but obviously we're still behind in terms of getting that research out into practice. Once practices have started changing, then I think we do have opportunities to go further, to actually understand why, for example, is rhizopogon a much better mycorrhiza than some of the other species we have. What is it doing different? And through that knowledge, then we can look to advance even further and make our nursery systems even better. Right, well, obviously a lot of people have been involved in this research. I have to mention Katrin Wilbert, Caragao, Sarah Edison, and Rodrigo Osorio in particular for their efforts and endless time under the microscope. And also Sarah Edison to make our life a lot easier, because she brought in the molecular techniques that allowed us to shift from morphological analysis to molecular analysis, which has made things a lot more quick. There's been a lot of brave nursery managers who have let me do crazy things in their nurseries. That again, I'd like to extend my thanks to them, but also to Jody and the FWPA to provide this opportunity to talk with you. And I'd also like to thank you for your attention. So thanks very much.
Great, thanks very much, Simeon. We've got a few questions here. One from Angus Carnegie. Do you actively inoculate with mycorrhiza in nurseries or rely on natural infection?
Right. So with regard to that question, we're generally using the mycorrhiza that are present within the nursery. In nursery soils, there tends to be a lot of mycorrhiza already. I know that there are a number of nursery managers who have tried commercial products. Mostly they're rubbish, frankly. They have inoculum densities, which is sort of like maybe a propagule per meter squared, and you're trying to establish that in an area and into a soil that would maybe have a few, hundreds of millions of ectomycorrhizal fungi within a meter squared already. Some nursery managers sort of create a witch's brew where they'll collect up a lot of fruiting bodies and make a mix and spread that out. But by the by, we're just operating with what's there. We're trying to see what is the sensitivity of the mycorrhiza that are already established within that nursery soil.
Okay, from Ben Bradshaw, to extend this question, were the ectomycorrhizal species used native to New Zealand or likely originate from overseas sources? What's the risk of introducing non-native ECM?
Right, I actually did a bit of research on this a while back, because in New Zealand we have a huge array of North American mycorrhiza spread throughout New Zealand. And this is a legacy of the fact that when these exotic conifers were introduced to New Zealand, they weren't introduced as seed, they were introduced as live seedlings that carried soil and the mycorrhiza with them. So basically you had live seedlings moving from North America to Kew Gardens, from Kew to Sydney, and then from Sydney into New Zealand. And basically we've plotted this out and we've traced how these mycorrhiza came into New Zealand. So basically we work with exotic mycorrhiza. We do know Pinus radiata can associate well with a number of native ectomycorrhiza, but Douglas fir, for example, doesn't. It has to have those North American, it has to have the right mycorrhiza with it, which is not our native New Zealand mycorrhizal community. In terms of the risk of introducing non-native ectomycorrhiza to New Zealand, that's we've long passed that barrier. We're full of non-native ectomycorrhiza.
One from Wolfgang Drexler. Do you have any information or thoughts on the mycorrhizal effects for cuttings production and deployment as compared with seedlings?
Yeah, that's one of the gaps, is that we haven't done enough work with cuttings. We have done a little bit around it, and that's predominantly around the potential for ectomycorrhiza to help induce root formation in cuttings. I'm waiting on a trial to complete. It was interrupted, unfortunately, by basically a bit of the COVID lockdown, but we do have some interesting results in that direction coming through. But that's sort of, again, that's not a large-scale trial. That's just testing some opportunities. But in theory, the literature suggests that it should be a good option.
From Andrew Moore, any similar research done in Eucalyptus globules or nitens?
We haven't, but there is a reasonable body of work around mycorrhizal associations with these species that I've looked at out of China in particular. That's reasonably interesting, but I wouldn't say it's as comprehensive as the work that we've done around trying to manipulate it and then see what happens next. It's probably been more along the lines of just the ecological side of what is there, what can you find in association with them. That said, I've just been asked to contribute to a project that's received some funding to actually start looking at Bossostonia.
Okay, Danielle Wiseman says some work was done in WA on native eucalypts on rehabilitated mine sites in response to the previous question. And also, is there data we can see on the effects on seedling survival of managing mycorrhiza?
Well, yeah, we've got a ton of data on that for Pinus radiata in New Zealand. And I would imagine there should be data on that mine site restoration work. I'm a little bit familiar with that because I know someone who was involved in it, but more from sort of a bacterial perspective. So I would hope that there would be data on that for the mine site work, but we do have a lot of data, and indeed, hopefully, I will have a publication out on some of those outplanting results that I presented today. Well, hopefully it'll be accepted for publication later this week, as a couple of days ago I submitted what were hopefully the final revisions. But yeah, there is information on that, and there are legacy effects.
Okay, Ben Bradshaw's just shared a comment. Plenty of work done on eucalypts, but they must compete with native mycorrhiza. Perhaps an opportunity to exploit site-specific ECM, etc.
Oh yeah, I would absolutely imagine. I mean, the process that we've gone through with Pinus radiata, we've just done, it was Pinus radiata because that's what we work with. That's 90% of our estate. There's no reason why you couldn't put another species, another exotic species, or indeed some of our native species at the start of that process, just to understand how sensitive they are, how specific they are in their requirements for a particular set of ectomycorrhiza, or if they're like radiata pine, which is essentially promiscuous. It can associate with a wide range of mycorrhiza, but it's clear that some of them are much better than others.
Okay, has there been any research on seedling water balance with and without mycorrhiza?
Accidentally there has, yes. We've done work on Pinus radiata and its hydrostatic pressure, hydraulic conductivity under different environments. And the people who were doing that, I wasn't directly involved with them, but when I did start looking at some of the work, I realized that actually they'd accidentally set up a sterile and, well, basically a plus and minus mycorrhiza gradient within their work just by accident. So we're yet to untangle it fully, but there is something there, I think.
Okay, a couple of questions about work in container nurseries. Has there been any work done in container nurseries, and how does the approach differ?
So on work in container nurseries, it basically just doing some now. We worked in seed beds simply because that's what we started with and that's what we were extending. But at the back of, but there's always been the intention to apply our learnings to seed beds because, to container systems, because it's just as relevant. And it is showing to be just as important.
Okay, and another question: from Stephen Elms, how does the trichoderma research interact with this work?
Well, good question, and one that I'm very interested in. I am doing, I am involved in one reasonably large trichoderma research program. We're working with a range of trichoderma clones to look at their impact, but at the same time, I'm also building capability in to understand, does the presence of particular trichoderma alter the balance of ectomycorrhizal species? What does it do to total mycorrhization? I know that there is some stories that the presence of trichoderma will restrict or reduce the abundance of mycorrhiza, but hopefully, literally in about a month's time, I'll be able to get my first hard data on that.
Great, thank you. And any context for southern pines / pine Caribbean pine and their hybrids, burn versus no burn site prep effects on in situ field populations of ECM?
So as part of an LTSP, that's long-term site productivity study, we do have some published results showing the effects of site management and site preparation on the ectomycorrhizal population present at a field site, and we do see the potential for long-lasting and enduring effects that for Pinus radiata, they have basically lasted longer than the life of the rotation. So there is sensitivity. Now, I'm not quite sure any context for southern pines, or is that context? I'm not quite sure what the first part of that question meant, sorry.
Yeah, it says context. Ian, do you want to talk to that one?
Yeah, thanks, Simeon. A very interesting professional presentation. Yeah, just the same principles, whether you're aware of any people doing research in that, subtropical southern pines.
Not really. Yeah, the other people I know who are doing work on this, there's actually a lot of people doing work on eucalyptus in other parts of the world. But no, I don't know much about people doing work on other pines, unfortunately, other than some older work done on Scots pine and some Scandinavian work as well. But yeah, nothing desperately recent of those. But
yeah, I was interested in we're 100% container nursery, and I was interested in your comment on the lack of benefits in applying any sort of powdered ectomycorrhiza, endo-ectomycorrhiza.
Oh, it's not to say that. Well, yeah, my approach has been that the mycorrhiza are there; they are adapted to that environment. Bringing in new mycorrhiza, they're going to have to compete for that environment, or you're going to have to provide them with a specific niche that gives them a competitive edge. Yeah, have always been a bit nervous about the approach of introducing a wholly new microbe into an environment, because generally what I've found is the capability is there. It's just encouraging the species or the performance or the activity that you want to see.
I guess, but what's the native mycorrhiza that's there when it's a fully containerized nursery, starting with a just straight out bark potting mix?
That depends on your practices and what's around you. I mean, mycorrhizal spore sources are everywhere. Basically, they're all over the place. We did some work in New Zealand, basically understanding, because that new ground syndrome that I sort of pointed to as being still a problem up to the 80s, that isn't an issue anymore. You can put Douglas fir down pretty much anywhere in New Zealand and it will grow, because there are enough spore sources around New Zealand now that the mycorrhizal fungi have spread, and they're basically everywhere now. And if you're starting off a container system, it's quite easy to get to the mycorrhizal spores from the previous crop will reinfect the next one. Or if you've got any trees or indeed suitable fungal communities in the soil anywhere near where you've got your container nursery, you could see those mycorrhiza coming in. Now, in terms of if you're starting a nursery from complete scratch, then you could do the trick that was used to defeat new ground syndrome. Go to an established effective plantation, go get some forest floor litter. It will contain mycorrhizal spores. You have to be careful though, because it will also probably contain pathogens as well. But see if you can add what you want, basically, or add the things you need.
Yeah, thanks for that. I guess I mean, we have our nurseries in the middle of a plantation, but I was just wondering whether we get an extra boost, if you like. Yeah.
Oh, absolutely. If you're in the middle of a plantation of the same species, that's your spore source. You're covered.
Yeah. Okay. Thanks.
Okay, and Angus Carnegie's popped a comment up there. New South Wales DPI Forest Science with Forestry Corp and Western Sydney Uni have been doing some preliminary investigation of mycorrhiza in Pinus nurseries, composition and root colonisation in different nurseries, glasshouse trials, looking at different mycorrhizae treatment, which have been planted out in nearly 1 year old, sampling of plantations for mycorrhizae composition. Ian, we have found pine mycorrhiza colonising containerised nurseries naturally. Yeah.
Oh, excellent. Good to see that. Yep, that they're doing work within this space. I mean, looking through the international literature, there hasn't been enough. There hasn't been enough work trying to say, well, here's what we do in the nursery, and here's what happens next. That linkage just isn't made often enough.
I guess one of the benefits I was thinking about was even if there's not a long-term response in the growth in the field, if the artificial inoculation speeds up the development of mycorrhiza in the container nursery, then they develop quicker, and you can sort them quicker and have a higher success rate in sorting. So better utilisation out the nursery gate. That's sort of where I'm coming from.
Yeah, no, there will be all sorts of opportunities. Like I say, I haven't really explored the opportunities in container nurseries in any depth. We're only just starting to look at that, really. But really, our focus is saying we have these opportunities in seedbed nurseries, which is still the vast majority of Pinus radiata production in New Zealand, and will be for decades more at least.
Yeah.
We can be doing better.
Yeah, thank you.
And some of the advantages of doing better is it's actually we're spending less money to produce what is a better quality crop. And apparently what seems to be an enduringly better quality planted forest as well, it intrinsically comes with greater risks. So we just need to improve our science so we can decrease that risk.
Ian, it's Angus here. Some of the preliminary results we've got indicate that some of the commercial mycorrhiza mixes you can buy aren't necessarily great for Pinus specifically, even though they might have a mix of species in there.
Yeah, thanks for that, Angus. We used one. We started a few years ago, but we haven't done any testing, serious testing to get any control versus treatment figures. So it's just interesting feedback. Thanks.
Okay, well, I think we've come to the end of the questions and we're a bit over time. So thank you so much, everyone, for joining in that and participating. And in particular, thanks very much for Simeon for that great presentation again. Hope you can join us all again this time next Friday for another presentation from Simeon about soil mycorrhizae, reducing stress.
Bacteria more this time than mycorrhizae.
Okay, great. Thank you. All right, well, thanks everyone. We'll see you again next week. Thanks very much, Simeon.
Cheers.
Thanks, Simeon.